start my talk with a video to tell you about what I work on. You're looking at the nuclei of a female cell and the two X chromosomes. And this is a natural phenomenon which happens in every female cell in the nuclei, where one of the two X chromosomes is chosen at random and it is inactivated. So there is no transcription from this X and no gene product. The reason this happens is because a normal human cell has 23 pairs of chromosomes, including one pair of sex chromosomes, the X and Y, where X is large and gene-rich, Y is small and it's gene-poor. A normal female has 2 copies of the X chromosomes. Males have only 1 copy of the X and 1 Y. You can appreciate from this that there is a gene dosage imbalance for these X genes between XX female and the XY male. A whole chromosome gene imbalance is either lethal or it has severe health consequences. For example, individuals with an extra copy of chromosome 21, this is one of our smallest chromosomes, develop a disorder termed Down syndrome, where they have severe mental retardation amongst other physical and health problems. So then it's not surprising that a mechanism has evolved to balance this gene dosage difference between male and females. This happens in human and other placental mammals by inactivating one of the 2 X chromosomes in every female cell. It happens early on in a female life, this is before implantation, and this inactivated X chromosome can be seen as a condensed heterochromatin body under a light microscope, and it's been termed bar body. At a molecular level, this mechanism is controlled by a master regulator gene called XIST, X inactive specific transcript, which is located on the X chromosome. This gene codes for an RNA, which is a functional non-coding RNA, meaning it's not translated into a protein. Instead, the XIST RNA codes the X to be inactivated, creating a clot over this X chromosome that brings about the inactivation machinery. Modifying histone, adding methyl groups ensure that you have a chromosome-wide, complete and stable silencing. However, some genes do escape inactivation. Human and placental mammals are not the only mammals with an X inactivation mechanism. Marsupials, these are the pouched mammals, the cute and cuddly tamawallaby and kangaroos, also have differentiated sex chromosomes X and Y, and they have an X inactivation mechanism. Our understanding of the marsupial X inactivation is somewhat limited due to the small number of studies in this field. So most of what we know is based on 5 genes and in 7 different species. What we know for a fact is that there is no XIST present in marsupials. Instead, they have a gene termed RSX, RNA on the silent X. This gene, similarly to XIST, codes for a functional RNA, which is not translated into a protein. Similarly to XIST, RSX creates a clot over the X to be inactivated. This is a female opossum nuclei, and here you're looking at the XIST clot in a mouse female nuclei. What we know from these limited studies is that the X inactivation in marsupial is not as complete or stable as that of the placental mammals. And today in this talk, this is what I'm going to be addressing by looking at escape from inactivation from the marsupial X, hoping to look at more than just 5 genes, and then looking at promoter methylation. So to study escape from inactivation, we chose to use a methodology which allows us to look at expression at each individual nuclei. This is done by fluorescently labeling a probe, this is your gene of interest, and then hybridizing this probe to a nuclei to look at the expression of RNA. So if you're looking at an X chromosome gene in males, when they only have 1 X chromosome, you should only see 1 signal. When you're looking in females, since they have two X chromosomes, there are two possibilities. You either see one signal suggesting that the second allele on the inactive X is inactivated, or you would see two signals suggesting that there is escape from inactivation. Here you're looking at a result of a gene on the X chromosome labeled in red, and this is a male nuclei. And when I've looked at 100 cells, out of these 100, all of them have only a single signal. When we look at females, however, what we notice is that there is a percentage of nuclei with a single signal, and then there is a percentage of nuclei, for this gene only 5%, where they have two signals. So then what we've done is to look at a number of different genes located on the marsupial X chromosome. And here is the percentage of two signals, nuclei, so this side, for each gene. And as you can see, each gene has a different percentage of two signal nuclei. When we compare this, so this led us to believe that there is a characteristic frequency of escape, because if you repeat this experiment, even in different cell lines, you would see that each gene has the exact same frequency. So when we compare this frequency of escape in marsupial X in comparison to frequency of escape in the human X, what we've noticed is that there are greater number of genes on the marsupial X that escape inactivation at, say, 30% of nuclei in comparison to the human X. This is confirming that marsupial X inactivation is less complete than the humans. This led us to ask the next question: why is there this difference between marsupial and placental X inactivation? One of the agents which we have looked at is DNA methylation. The reason for this is because there is an inverse correlation between methylation and expression. Here you're looking at a transcription unit with a gene body, a transcription start site, and a promoter. Promoter is where the transcription machinery binds to, and if it's not methylated, then you will have transcription of RNA. However, in a situation that promoter is methylated, the transcription machinery cannot bind to it, and therefore there is no expression. When you look at promoter of genes in the human X, promoter of genes on the male X, and the active X in females are not methylated, and these genes are expressed. However, when you look at the inactive X chromosome in females, majority of genes which are subject to X inactivation are methylated. However, those genes which escape inactivation, these are the genes which are expressed, the two signal cells, you would see that they are not methylated. So we wanted to know if the same applies in marsupials. So we have done a genome-wide study looking at a 6 kb region. This is 5 kb upstream of the transcription start site into the promoter to cover the core, distal, and proximal promoter, and 1 kb downstream of the transcription start site into the gene body. We are looking at male versus female. The reason for this is because I cannot distinguish between the active and inactive X chromosomes. Therefore, we are using the male-only X as a proxy for the female active X chromosome. So I'm going to show you results of a female-to-male methylation ratio. If there is no differential methylation between female and male, then you would expect this ratio to be equal 1. However, if there is female-specific methylation, this ratio is going to be greater than 1. Since we've said that the active X and the male X are identical in their methylation pattern, this female-specific methylation should be on the inactive X chromosome. So here you're looking at that 6 kb region of interest, and this is the female-to-male methylation ratio for all the genes on the marsupial X. And what you would notice is that there is a female-specific methylation around 2 kb upstream of the transcription start site. This methylation has an inverse correlation with expression, although I am not showing you the data at this talk. What is interesting is that the methylation which has been associated with placental X inactivation is much closer to the transcription start site and within the first 500 base pair. What we would like to know is if this is marsupial specific or if this -2 kb methylation in female also is present in placental mammals. Also, what would be interesting to find out is if there is a correlation between RSX in blue and genes which are transcribed and genes which are not transcribed. So what we would like to do is to do a pull down of RSX to see if RSX specifically binds the methylation machinery. But those genes which are depleted of RSX can't bind the methylation machinery and therefore they are expressed. So to bring this with today's theme, how can the knowledge of exist RSX and other species but humans help us better off tomorrow? You would have noticed that the natural function of exist is to silence an X chromosome. We can take advantage of this function and maybe silence the extra copy in individuals with Down syndrome. This is an extra copy of chromosome 21. This has been performed on cells derived from a patient with trisomy 21, and exist has been inserted into these cells, and they have successfully shown that you can inactivate one of the extra chromosome 21s, which is shown here as a bar body. The problem, apart from still not being able to transfer this into an actual individual, is that you get escape from inactivation. So although you are inactivating a whole chromosome, there are still some genes which are being expressed. How could marsupials help us? Well, no one has tried to insert RSX into a system like this. RSX has the same function, but it's using different molecular agents to reach this final inactivation. So maybe having RSX and exist together into a system like this could give us a better silencing. Trisomies are not always based on just a whole chromosome. Sometimes there is only a smart fragment of chromosome which has been translocated onto a different chromosome. So even if RSX doesn't shut down a whole chromosome, what it could do is to shut down a smaller region, and it could be used for these sort of translocation trisomies. Of course, marsupials are not the species with an X inactivation mechanism. You could look at other long non-coding RNAs from species such as platypus and chicken, which also have a dosage compensation mechanism, to see if we could use them as potential therapeutic agents. With that, I would like to thank all my colleagues and collaborators, and I would take any questions. Thank you.